again, it's just the background.
Just
So.
everybody. Thank you for attending Alyssa Strickland's dissertation seminar. I wanted to tell you just a little bit about the history. So she came here as Alyssa Stacy with a BS in genetics from Clemson. Give you a timeline here. She applied in 2019, says she wanted to do population biology and genetics. Then came here with the MS in September of 2020. Then COVID happened. March of 2020, she had her proposal. And in November of 21, she defended her thesis on oyster larvae distribution, working with our HAP program, and decided that she wanted to be punished a little bit more. So she applied to be a PhD student here, came here in February of 2022, went off and had a wedding, and now it's June 25 and she's defending the dissertation. So she has done what she wanted to do, population biology and genetics, even though she took a little bit of a detour, but you had to do that to figure out what the ecology part was. In her spare time, she goes on nature walks with the dogs. She fosters dogs, but probably more important than the dogs is she likes to go scuba diving with Carl. And she's going to have a postdoc in NC State that starts in July
1st.
Yep. And the problem is that she has been our mapping genius for every paper we've written since I got here. And none of us know how to do what she does with mapping because it doesn't work on any of our computers. I think she like put a bug in there that says, if the computer is Alyssa's, this will work. So I don't think you're going to see any more maps in any of the papers that come out of our lab once she leaves. So Alyssa, please. The maps at the back. He said, train us a little bit more on what you've done.
All right. Well, thank you everyone for coming today. My name is Alyssa Stacy Strickland, and today I'm going to be talking about my dissertation work, which is titled Eastern Oyster Genomics in a Well-Mixed Estuary System, examining population structure, viability, and response to stressors. Oh, I forgot I have to do this. No, my thing isn't even working right now. There. Before I get started, I just want to mention that this dissertation is dedicated to my husband's grandfather, Dick Pesta. He passed away last year, and he completed all but his final steps of his own PhD journey while also working a full-time job with 3 kids. And so I know he's proud of us and looking down on us. We wish we was here. To get started, let's talk a little bit about Eastern oyster history and why we are studying these organisms. They were discovered in the 17th century. When English colonists came over, they discovered hundreds and hundreds of oyster reefs along the coast, and the value of oyster was then recognized. They would ship them out, and they were kind of like the common food, how lobster was the common food back in the day. However, fishers in the 19th century began to start recognizing the importance of oyster farming and how lucrative it could become. So this led to the start of the traditional oyster farming that we know. As you can see in the picture over here, we used to have sailboats that would have a huge net on the bottom and dredge up the oysters along the bottom. And while this was great, they were getting a bunch of oysters. This caused huge population declines. Those declines were also exacerbated by climate change, overharvesting, and introduced diseases, which can also be attributed to climate change. Unfortunately, all of these have led to really drastic declines in the population. In 2018, oysters accounted for 30.4% of all mollusk aquaculture and accounted for over $130 million in sales. In Great Bay Estuary, we're seeing the native population decrease with just over 40 hectares of native oyster reef in Great Bay. We're not seeing native populations thriving as they once were here. At the same time, there has been an increase in oyster aquaculture within New Hampshire. In 2024, there was over 1.2 million oysters harvested there. There is just about 40 hectares of licensed oyster aquaculture sites. There was about $6 million in economic benefit of the oyster aquaculture industry in New Hampshire, and a 553% increase in the value between 2014 and 2024. So why are we studying these organisms and why are they important? They have 3 main features that we can classify what they do for the environment: nutrient cycling, habitat creation, and shoreline protection. A single healthy oyster can filter up to 50 gallons of water a day, aiding in water filtration and nutrient cycling, especially in carbon and nitrogen. They also provide a habitat for juvenile fish and marine life, such as smelt and trout in Great Bay Estuary. Oysters cluster together, so they settle on each other when they're babies, and then they grow on top of each other and form clusters that are called oyster reefs. They kind of mimic coral reefs and estuary systems, and so the fish are able to hide in those oyster reefs. The final thing is that they aid in shoreline protection. So in one study done by Brandon et al. In 2016, they ran Hurricane Sandy simulations, and the results showed that the removal of the oyster beds right directly offshore decreased the wave energy between 30 and 200%. Since the major theme of my dissertation is using genomics as a tool for studying oysters, I'm going to kind of bring us back to high school biology and talk about central dogma. The central dogma describes the way in which information flows from DNA to DNA through replication, DNA to RNA through transcription, and RNA to protein through translation. DNA is the genetic material of all organisms on Earth and is divided into functional units called genes, which provide information needed to generate the functional protein. To think of this in a little bit of an easier way, imagine DNA as the blueprint for a house. The RNA is the copies of the blueprint that we are putting notes on. We're putting more detailed measurements and stuff on. And then the protein is the actual house that is being built. In this dissertation, I'm using Oxford nanopore technologies for a majority of my work for multiple reasons, including the ability to sequence RNA directly, to sequence ultra-long DNA, and for real-time analyses. So to go over Oxford nanopore, the nanopore that is in the chemistry is inserted into a membrane that's created by a synthetic polymer that has a high electrical current. The current is generated through the nanopore, and then single molecules can pass through the nanopore and cause disruptions in the current. These include DNA, RNA, and proteins, and depending on the size or the disruption that is passed through, we can get a different signal which is attributed to different materials. The current disruption is measured and the molecule can be identified. So then the nanopore processes whatever fragments are presented to it in real time, regardless of the length, which is different than traditional sequencing here. And this can be done at an exceptional speed, as you can see here. In the age of advancing genomics, marine invertebrates, specifically oysters in this case, have benefited from genomic tools. These two studies here, which at the top is the sequencing of the genome of the eastern oyster and the development of a high-density SNP, or single nucleotide polymorphism, which is just a single base pair change that we use for genetic differentiation tests. An array for the eastern oyster there have been used as a backbone to allow for further analyses to determine population differences, etc. Some of these, as I just mentioned, include population genomics. So here you can see several papers of population genomics done in different estuary systems, as well as gene expression analyses in response to temporal conditions and disease pressure. And then the last thing is they're being used for aquaculture development to create more tolerant oysters. So these tools that I discussed and the previous studies have allowed for the creation of this dissertation. In general, this dissertation aims to build off the studies that were previously conducted by using multiple genomic methods to study eastern oysters in Great Bay. With these new novel methods, I hope to address causes for population decline and refine methodology that's currently used in Great Bay Estuary for oyster restoration. Specifically, I used population genomics to study the population structure of oysters, extracted ultra-long DNA from oysters and compared it to the established reference genome, used gene expression to understand differential response to stressors, and identified long non-coding RNAs that are differentially expressed in varying oyster groups. To get started, the first thing that we needed to do was understand how the different oyster subpopulations were acting in Great Bay Estuary. Previous studies have shown that Great Bay Estuary, we think, is a well-mixed estuary system. We're not so sure about that now. It might be bilaterally mixed. And so we wanted to see how the oysters are behaving and how the populations are differentiated between this ecosystem. Population genomics is a powerful tool. It's a common analysis that can be used to better understand variation within populations. For example, if we were conducting a population genomic analysis on this oyster reef right here, we would take all the genetic data from each individual and then look at it in one group to try to infer population level analyses. Results from population genomic analyses are extremely important to consider for management strategies, as ensuring genetic diversity increases a population's fitness or ability to adapt to change, and then thus further prevents mass extinction events. Population genomic analyses have been conducted on eastern oysters and other estuary systems, as well as Great Bay, and have shown strong population structure on a fine scale in these other estuary systems. And since Great Bay is well mixed, as I mentioned before, comparing population genomics of Great Bay with other estuaries could provide interesting results. Therefore, the goals of this chapter were to use population genomics to identify population structure and differentially express single nucleotide polymorphisms, or SNPs, as I mentioned before, of oysters in Great Bay Estuary. So to do this, I collected a bunch of oysters, 210 in total, from varying oyster sites within Great Bay Estuary. There were 4 native sites. Those are the blue dots that are seen here. And I want to make note of this special site, Adams Point, right here. All of the native populations, except for Adams Point within Great Bay Estuary, are subtidal, and Adams Point is this intertidal population. I then also collected oysters from 2 cultivated sites within Great Bay, and then the restoration site that we are currently working on within Great Bay Estuary. Then I extracted a piece of the mantle tissue and conducted whole genome sequencing, low coverage at about 5x. Once the sequence data were obtained, I ran through a series of filtering and various analyses. This slide is more for my committee, so no one really needs to pay attention to the details of this. So there's a lot of terms on here that might not make sense to you, but overall, what I did was use various genetic tests to determine the genomic differences between location samples and to see if there was any clustering present. Anything that I do mention without this, I'll explain in more depth. So to kick off the results, this first plot over here on the left shows a discriminant analysis of principal components, or a DAPC, and the one on the right is an admixture analysis. A DAPC is a statistical technique used to analyze complex data and identify groups or clusters within that data. Here, I used it to generate clustering data based off genomic profiles of the oysters I sampled. So each dot here is representative of one individual's genomic profile, and the color is corresponding to the site that they were collected at from the map that I showed earlier. So to understand this a little bit more, imagine that you have a large collection of different colored marbles. A DAPC would then be an automatic way of clustering those models into the right, or those marbles into the right color without you having to do the work, and then tells you how similar those marbles are to each other. Here we're seeing that the native sites, so those ones in blue, are clustering together, but we are seeing that the subtidal populations are clustering separately from the intertidal populations. We see the native population over here, or the cultivated population up here on its own, and then interestingly, we found that the restoration site was over on a cluster on its own. We didn't expect to find this, as the restoration site is on a dead native reef, and we are using cultivated oysters to help supplement the population. So we expected to see kind of a mix of the native and cultivated populations, but we're finding that it has its own genomic signature. To look at the admixture analysis, so this picture on the right, these analyses disregard which location and heritage each sample came from, and then uses modeling tools to assign a cluster for each of the samples. The model suggested that we use 3 clusters here, so cluster 1 in red, 2 in blue, and 3 in yellow. And interestingly enough, we found that the native populations clustered into cluster 2, the restoration population in cluster 3, and the cultivated in cluster 1. We did see influence from all individuals or all clusters on each individual itself, which does support that Great Bay Estuary is well mixed in nature. There is some mixing going on that we're seeing all of the different clusters in each thing. To see this on a map by the sampling location, this map shows the proportion of genomic influence of each subpopulation at each site. So again, this is just taking this data up here and translating it onto a map so you could see it better. I then conducted a genome-wide association study, which identifies SNPs that are differentiated between 2 groups to see if there was any significant SNPs between cultivated and native populations in Great Bay Estuary. These results hopefully will be used to create a genomic test to determine heritage of spat found in the wild, so native or cultivated, so we can determine which populations are contributing to that restoration pool. We found 145 significant SNPs or single nucleotide polymorphisms. As I said earlier, those are just one base pair changes in the genome that we use to determine genetic differences. Those were all found at a p-value of less than or equal to 0.05, and 76 of those were in coding regions, which means that those are located in regions that translate to proteins. From these results, I found a few interesting takeaways. First, I found that genomic separation between the native and cultivated oysters, which we expected to see. And interestingly, we also found separation between those two groups and the restoration effort, or restoration oysters. As I said previously, this is interesting to see at the restoration site as we have the cultivated oysters on a dead reef. And so what we expected to see was mixing between those two populations rather than its unique genomic signature. Additionally, I did see some separation between the intertidal and subtidal populations of the native oysters, and this warrants future studies to potentially identify genes that are present in each group and could help with more tolerant temporal gradients. I did find quite a number of significant SNPs that were different between the cultivated and native oysters, and future work that we are currently working on right now will determine the heritage of spat at that restoration site to determine or to kind of evaluate the genomic influence of the recruits that we're finding. To further this a little bit more, we can work on creating a more streamlined genomic assay to test the genomic heritage of spat in Great Bay Estuary. So now that I obtained the population level data, the rest of my dissertation aimed to use newer genomic technologies to understand oyster populations a little bit more in depth. The second chapter aimed to successfully extract and sequence ultra-long DNA from mucopolysaccharide-rich eastern oysters. Recent advantage in technologies generated by Oxford Nanopore Technologies, which I showed you a diagram on before, have allowed for the sequencing of ultra-long DNA since it's not size restricting. And this ultra-long DNA is DNA that is greater than 50 kilo base pairs. This is extremely useful as it can be used in combination with reference genome to create a complete genome without any gaps or a telomere to telomere assembly. This allows for improved understanding of complex regions, identification of previously hidden genetic variants, and a better annotation. Unfortunately for us, oysters are coated with mucopolysaccharides, which are long linear polysaccharides consisting of repeating disaccharide units. That was the technical term. That was just for everyone that needed to know that. Essentially, this is all of the goo that you see within an oyster. It's what makes it slimy, and if you're eating an oyster, kind of slide down your throat better. Extracting DNA, though, from mucopolysaccharide-rich organisms is very difficult, as to remove the mucopolysaccharides, you have to do a lot of shaking and vigorous activity with the DNA, and so that breaks up the DNA and shears it into really small lengths. So it doesn't give us the ultra-long DNA that we want. However, if the methodology can be created to remove the mucopolysaccharides but preserve the ultra-long DNA, this can be useful in combination with the existing reference genome. Because of this, I aim to extract ultra-long DNA from the mucopolysaccharide-rich oysters, sequence the ultra-long DNA with Oxford Nanopore, compare the generated data with the existing reference genome, and then classify everything that was left over after the genome assembly and alignment, as you'll see later, disease and bacteria DNA. So to do this, we collected hemolymph from the oyster adductor muscle, as you can see in this figure, and also my lovely undergrad, who is no longer an undergrad, Kaylin, doing it right there. Hemolymph is a fluid found that's in vertebrates, particularly mollusks, that functions similar to blood in vertebrates. It circulates within their body, directly contacting the tissues. It contains hemocytes, which are cells that play a crucial role in the immune system of these animals, and also some bacteria and disease-causing organisms like MSX and dermo. Oysters have an open circulatory system where the hemolymph flows from the heart to the peripheral organs and the body cavity, where the tissues are surrounded then by the hemolymph and then diffuse back into the circulatory system between the cells. I then used ethidium bromide wash solution to remove the mucopolysaccharides through a series of wash, gentle agitation, and spinning down. The ethidium bromide intercalated into the DNA and protects the DNA during extraction, allowing for the removal of the mucopolysaccharides. I then did ultra-long DNA extraction following the Oxford Nanopore guidelines, and then prepared it for sequencing in a Promethean 2 Solo, which is ONT's device for sequencing. To kind of visualize this, when we think of DNA, we think of just like clear fluid in a tube. We don't see any DNA. Ultra-long DNA is so abundant that we actually see the DNA here. So this is DNA from a ultra-long DNA extraction. And then as you can see here, I'm loading it into the flow cell of the P2 Solo, and it's so thick that it doesn't flow through at a continuous rate, so you have to do it drop by drop there. After the ultra-long DNA was generated, I assembled the reference genome without a reference or de novo, so just by itself, using a program called HiFiism that's used for long-read genome assembly. This produced 105 contigs, or basically chunks of DNA, and with it, that had a GC content of 34.62, which was comparable to the established reference genome. When I compared the established reference genome to my de novo assembly, it accurately assembled only 57.6 of the genome and had 543 misassemblies, which was not great. Was not happy with those results. But to visualize this alignment along the established reference genome, here you can see a plot where each of these dots is a forward or reverse alignment, and this is the eastern oyster chromosome, just kind of plotted out onto the x-axis. They have 10 chromosomes, and so each little, you could think of it as a line here, would be each individual position. In addition to this de novo assembly, we also mapped the reads to the reference genome and then looked at the unmapped data to see what it was compromised of. When we mapped to the reference genome, only about 5% of the reads were classified as unmapped, which was very good to see. We then ran the unmapped reads through a metagenomic analysis, which is a study of all the genetic material of all organisms in a sample. To think of this, think of all of the books in a library are each organism, and we take the pages from each of those books, shred them up, and then try to identify which book corresponds to which shred based on little parts of the book that we know. And this is kind of metagenomic classifying in the very simplest of terms. So this plot here shows the unmapped reads that were not eastern oyster DNA and the percentage that mapped to the species found. We found a majority that were bacteria here, as well as some human DNA, which is consistent with wastewater studies that have been done in Great Bay Estuary. None of them mapped to the disease-causing organisms that we expected to see. However, some of these are known oyster pathogens, such as Vibrio, which would be somewhere in here, but it's not shown on this plot. The most exciting thing about this was that we were able to get ultra-long DNA from that mucopolysaccharide-rich oyster, which was very hard to do and took probably 20 different experiments for me to do it. However, the de novo assembly produced not the best results, which can possibly be explained by extensive repetitive regions within the eastern oyster genome. Repetitive regions in the genome are estimated to be about 35 to 54% of the total genome, and these are challenging for de novo assembly. So future research can use a different tool to assemble the genome de novo, one that can possibly help do better with extensive repetitive regions. Very minimal contamination DNA was found within the ultra-long samples. We didn't see evidence of the known oyster protozoan pathogens that we expected to see, but we did see some bacterial pathogens of oysters. We also saw evidence of DNA found in wastewater, which has been consistently an issue for Great Bay Estuary. There have been issues with the wastewater in the Lamprey River, which has migrated down into Great Bay as a whole, and so we hate it, but we see it all the time. In general, this research in this chapter provides a fundamental stepping stone for a complete genome assembly of the eastern oyster, or telomere to telomere, which can enhance future studies later on. Next, to study how oysters under varying stress conditions and from varying heritage are behaving, we conducted a gene expression analysis of these groups. Gene expression analyses are useful tools that are used to study the up or down regulation of genes or clusters of genes with related functions to provide insight into how organisms respond to varying conditions. Gene expression is a process where the cell reads the instructions in a gene or a segment of DNA and then uses them to make a product, which is usually a protein. To think of this in a different way, you can think of it like following a recipe where the recipe is the gene to create a dish or to create a protein. The dish is what we can see, which is the phenotype, which the protein produces the phenotype, and the gene expression is basically telling us how much or how little of an ingredient to use. In oyster and oysters, gene expression studies are critical for understanding response to the environment as well as disease exposure and tolerance. As you can see in the top two photos, these are native oysters that I've collected within Great Bay Estuary that I've sampled. They are huge, sometimes bigger than my hand, and all various shapes and sizes. However, the picture below it is New Hampshire cultivated oysters, which they're all pretty, just generally more appetizing to look at. And so we know that there are differences between these two groups. A lot of these studies are conducted in laboratory settings where all the conditions can be controlled and normal environmental stressors and fluctuations are not present. Therefore, taking what we know from the laboratory experiments and combining it with field data can prove useful for the enhancement of eastern oyster gene expression studies. Because of this, we used a field study to identify differentially expressed genes, or like I mentioned earlier, the more or less amount of ingredients we needed in a recipe, to examine differences between cultivated and native oysters, as well as between oysters under varying temporal conditions. After that, to understand what the genes are doing, I briefly, very surface level, examined the roles of these genes to see how they can account for different conditions. To do this, we collected 5 cultivated oysters from 2 different sites within Great Bay and 5 native oysters from 2 different sites, and we did this once in July and once in August. Again, a piece of the mantle tissue was then excised, and we prepared DNA or RNA for Oxford nanopore cDNA sequencing. Here are all the technical terms of the things that I did, the analyses and the steps that I took. I'm not going to go over them too much, but generally, I sequenced the RNA, identified known genes based on the reference genome, looked at changes in the expression between the two groups in each comparison, and then looked into the processes associated with these genes. I ran 6 different experiments. I'm not going to talk about all of them. I'm only going to be talking about the experiment looking at gene expression between cultivated and native oysters, and then looking at oysters collected in July versus those collected in August. When discussing collection months, so July or August, we had one sampling date in July shown with the red box labeled July 22, and one in August labeled August 6th. In July, we had across all sites a higher temperature and lower salinity than in August, which allowed for a great comparison between the two groups. Note that where the salinity line in panel B for the Lamprey River has a 0 reading there, the data logger that we used went out, and so we weren't able to generate salinity data for those days. To kind of work around this, we averaged out the last known data that was logged and the data that was logged immediately after it went out, and then generated a line based on that. So I'm going to go over the results right now, and these figures have a lot of information packed into them. So just kind of bear with me as I explain it, and then we'll be seeing this again in the next slide. So just remember what I'm saying for this. Altogether in this figure, we measured which genes were turned on or off in cultivated oysters versus native oysters to see if heritage affects gene expression. In panel A, each of the columns here on the bottom is an individual oyster, and each row corresponds to one of the top 30 differentially expressed genes. The colors represent how strongly each gene is turned on or off. So yellow or orange in this case is high expression, and purple is low expression. You can see clear differences in color patterns between the two groups and very clear clustering between cultivated and native populations, suggesting that many genes are expressed differentially depending on the oyster's background, which we expected to see. Panel B here shows shows all the genes that are significantly differentiated between the two groups. Each dot represents a gene that is differentially regulated between the two groups. The x-axis shows how much the gene expression changes between groups. The y-axis shows how statistically significant the difference is. So the higher they are, the more statistically significant they are. The red dots show ones that are statistically significant and also differentiated highly, which can be used for future research on determining candidate genes between the two groups. You can see that there are many genes showing strong differences between the expression, meaning that these two groups have distinct biological activity. Panel C down here shows a summary of overall gene expression patterns, where each point is an oyster sample. Samples that are closely together have similar gene expression, while those farther apart are more different. Blue dots are native oysters and red dots are the cultivated oysters. They form two pretty distinct clusters here, as you can see right kind of down the diagonal, which we kind of saw in the first two plots and reinforces what we've been seeing. Together, these figures show that cultivated and native oysters express many of their genes differently and suggest that oyster heritage has a big effect on how its genes behave, which is what we've been expecting to see all along. To now look at this with collection month, this figure is similar to the last one. However, here we measured which genes were turned on or off of oysters collected in July versus those collected in August. In panel A, you can see some mild clustering between the two groups. However, we don't have the clear-cut clustering that we saw between cultivated and native oysters. Panel B shows the genes again that are significantly different between the two groups, and we're seeing a lot fewer genes here that are significantly differentiated. And again, panel C shows the summary of overall gene expression patterns, and as we saw in the previous two plots, we're not seeing any distinct clustering between July in green and August in gray. Together, these figures show that there is not as clear separation here between collection month under varying salinity and temperature conditions. This plot is a summary of all 6 of the experiments that I conducted together and shows the functions of the genes that we found. We see the biological functions that are enriched in genes showing significant expression differences across all of the comparisons that were conducted. This is kind of just a high-level view of what is going on within the genes. In this figure, each of the rows is a biological function that was assigned based on one of the genes. The x-axis shows the strength of the statistical significance. So as we move farther away from the y-axis, they are more statistically significant. The size of the dots is how many genes are involved with that function. The bigger the dot, the more genes are involved with that function. The shape and the color of the dots tells you which group or comparison it came from. So overall, this figure summarizes which biological processes are most affected in the different comparisons that were studied. Functions such as ribosome assembly, RNA binding, and nucleotide metabolism repeatedly show up as significant, most likely meaning that they are conserved as a biological function in response to these stressors. To view this in a different way, I then created a word cloud of the functions where the larger words correspond to a higher amount of genes involved, and a lighter color is a more significant p-value. And so this provides the visual summary of what we're seeing on the left-hand side without having to look at which experiments are being conducted. Terms like nucleotide binding, organelles, ribosome, and metabolic processes are larger, which means that these functions have more genes associated with them across all experiments. Now that we've explored the genes differentially regulated and found out what they mean, what does this all mean and where do we go from here? So we found out that heritage plays the most important role, as expected, and drives response to environmental pressure. We expected to see this as we know that cultivated oysters are selectively bred to tolerate disease pressure, as well as varying temporal conditions, while native oysters are not. And so that is why we are seeing these differences in gene expression. The strong differentiation and large number of genes in this comparison support a long-term divergence of these populations despite sharing the same well-mixed estuary system. Although there was high variation in the number and identity of differentially expressed genes across comparison, several functional categories were central to all comparisons, suggesting that oysters have underlying biological processes involved in their response to environmental stressors, and these are more conserved across all oysters. The next steps to build on this research would be further investigation into those biological processes affected by the differential gene expression, as well as examining the role of epigenetics on these genes, as it's known that epigenetics can play a role on gene regulation. Speaking of epigenetics, to segue into this next chapter, we know that epigenetics can affect gene regulation, so this chapter aims to look at one type of epigenetic modification, long non-coding RNAs, and identify them and examine them under different stressors and heritages of oysters. So long non-coding RNAs, or as you will see, LNC RNAs, are non-coding transcripts greater than 200 base pairs, meaning that they don't code for specific genes, and they have been increasingly studied and shown to have roles in a numerous amount of biological processes involving protein generation. Think of long non-coding RNAs as a manager, a director in a factory. In the factory is your cells, not the workers on the assembly line. Those would be the proteins, but they are the managers telling the workers what to do. Recently, these studies have shown long non-coding RNAs act as key modulators of response to stress and adaptation to environmental change, meaning that they affect genes greatly that are involved in the responses to these stressors. Although these long non-coding RNAs are well categorized in model organisms, their role in marine organisms remains largely unstudied. They're recently becoming more studied. However, we're lacking data on these marine organisms. Recently, a study on Pacific oysters found that long non-coding RNAs are associated with their response to heat stress, with more studies in eastern oysters. The role in eastern oysters has not yet been discovered, so because of this, since we know that long non-coding RNAs can fine-tune the response of an organism, studies into eastern oyster long non-coding RNAs are important and needed. The goals of my fourth chapter were to understand how native and cultivated oysters respond at a genetic level to stress and environmental conditions like changes in temperature and salinity. So to do this, we focused on long non-coding RNAs, which don't make the proteins again, but help control other genes and can provide those fine-tuned adjustments. To do this, we subsampled oysters from the previous chapter, and we used one native oyster from each of the two sites and one cultivated from each of the two sites, once in July and once in August. Again, we extracted the mantle tissue and then prepped the RNA for Oxford Nanopore native RNA sequencing. From the sequencing results, we identified over 67,000 transcripts. I then went through a series of steps to identify the differentially expressed transcripts and found 198 that were associated with the month comparison and 574 associated with the heritage comparison. We then went through some more steps, all outlined here that I won't go over, but basically selected for the size and then made sure that they were not coding so we could get long non-coding RNAs. After all of this, we ended up with 13 differentially expressed long non-coding RNAs for the month comparison and 32 for the heritage comparison. Similar to the chapter 3 results, these figures show clustering based on the genomic or the long non-coding RNA profile of eastern oysters. And so we can see distinct clustering just right down the middle between cultivated and red and native oysters in blue, meaning that they have different long non-coding RNAs. Again, for the collection month, July is in green and August is in gray, and we are again seeing the separation of the long non-coding expression profiles between these two groups. Again, similar to chapter 3, this plot shows the biological functions that each long non-coding RNA was associated with. There are fewer long non-coding RNAs shown here than we found, as a majority of them had no known function, and so I wasn't able to plot those out. The larger dots, again, mean that more long non-coding RNAs were associated with that function. The green dots here are coded to show the heritage comparison, or native versus cultivated, and the orange ones show the month comparison, or July versus August. Overall, this figure summarizes which biological processes are most affected in different comparisons. The functions found for collection month, so those in orange up here and then the last one down here, can be generally categorized as functions associated with oxidative stress regulation, while the other ones found for heritage, these green ones, can be generally categorized as innate immune functions. These two figures here, there's one figure on the left and one figure on the right. These are co-expression networks of the long non-coding RNAs that we found in the heritage comparison, that one on the left, and the month comparison over here on the right. Don't worry about paying attention to any of the names of the long non-coding RNAs. They are not annotated, and so we get this string of characters and numbers that really don't mean anything to us right now. This co-expression network shows how different long non-coding RNAs work together in oyster, like a social network of genes. They help find the long non-coding RNAs that are most connected to each other, or the hub long non-coding RNA. If you're thinking of these images as like a social network of genes, where all of these long non-coding RNAs are in one social network together, the hub, or the one that's outlined here and has stars there and star there, those are the influencers of that social network. On the left, when we're looking at heritage, We have two different modules or social networks shown. They are in gray and blue. The lines connecting the boxes mean that those long non-coding RNAs tend to turn on and off together, or generally more expressed together. The two long non-coding RNAs that are outlined in orange and have that orange star are that influencer of the group. And in this comparison, we are seeing that many of the long non-coding RNAs are working closely together. On the right, when we're looking at collection months, so over here in this light orange color, there was only one main module shown in that light orange color. The lines connecting them show the ones that are turned off, on and off together. And in this one, we saw that there was only one closely connected module, and the hub may be acting as a key regulator of the response there. So this research is super exciting as it's the first study of long non-coding RNAs in Eastern oysters and the first study of any epigenetic modifications of Eastern oysters. This is my husband before he was my husband, helping me in the lab during my master's when I was in the lab every Friday night at 8 p.m. and would cry and complain that I was there because I just wanted to be done, so he would help me run gels. When we looked at the heritage comparison, we found distinct clustering between the native and cultivated oysters, and the long non-coding RNAs had functions related to immune-related categories. We also found clear clustering for the collection month and long non-coding RNAs that were involved in oxidative stress regulation. Seeing changes in immune-related categories in oysters of varying heritage makes sense, as we know that the cultivated oysters are bred to be disease tolerant, so we would expect to see the differences in these immune-related functions. Similarly, the long non-coding RNAs involved in oxidative stress regulation make sense, as changes in temperature and salinity can induce oxidative stress. So together, this tells us that environment and background are both playing big roles in how the oysters regulate their genes. We saw through the co-expression network analysis that these long non-coding RNAs have networks that kind of talk to each other and help regulate each other. Depending on whether the oysters are native or cultivated, or depending on the collection month, different networks are activated here. Each network has its own influencer that could be crucial for how oysters respond to stress or stay healthy in different environments. These influencers could also be useful targets and could be able to be serving as biomarkers or biological flags to tell us how an oyster is responding to its environment. To improve the study, the sample size should be increased. We were extremely limited on funds and capacity to increase the sample size, so we did as much as we could. But we should be increasing the sample size and also combining these results with the gene expression studies to paint a clearer picture of gene regulation and how these oysters are responding to their environment. So to kind of summarize everything and bring it to a broader scale, this research uses cutting-edge genetic tools like DNA sequencing, gene expression analysis, and epigenetics to understand what's happening beneath an oyster shell, how they're responding to stress, where they come from, and how we can help keep them alive. The first chapter emphasized the restoration practices. They need to protect genetic diversity, or we risk rebuilding oyster populations that aren't as resilient or as fit and able to adapt to their population or their environment. The second chapter developed methodology to extract the ultra-long DNA from eastern oysters and use this in sequencing. Better DNA data mean better tools for breeding, monitoring, and understanding oysters at the molecular level. The third chapter found that oysters in Great Bay show gene expression plasticity. Knowing which genes are involved can help us select strains that will survive better in stressful conditions, especially in the face of climate change. Finally, Chapter 4 identified novel long non-coding RNAs in eastern oysters. These may hold a key to breeding oysters that are more resilient in the changing climate. To summarize this even further, this work lays a foundation for genomics-guided restoration that we can use in the field. These efforts should be focused on rebuilding oyster populations, not just by putting shell in water, but also by making sure we're providing healthy, genetically diverse oysters into this restoration population. This work can benefit from future studies with controlled experiments to test how oysters respond to heat, disease, and salt changes in a more controlled environment. Using these controlled field experiments would narrow down the genes and the epigenetic modifications that we saw. Additionally, we can use these data with ecological modeling as a guide for smarter restoration strategies. In summary, this work demonstrates that a shift towards using genomic insights can potentially help future-proof the oyster populations that we're seeing in Great Bay Estuary and provide a great genetic background for these oysters. Getting into my acknowledgements, I want to thank my advisor, Dr. Bonnie Brown, for everything and making me the scientist I am today. My committee members, Dr. Liz Harvey, Dr. Kelly Thomas, Dr. Dave Bouchek, Dr. Mitten Jane, Brianna Group, and Robert Atwood for all the help in the different areas of expertise you guys have. All of my funding sources, the UNH Ecogenetic Lab, Dr. Bo-Young Lee, Dr. Kelsey Meyer-Rust, Dr. Grant Milne, Tasha Sims-Harper, and Kaylin Grove, as well as all the past members of the Ecogenetics Lab who have helped me throughout this time here, as well, and then also all of the people who have helped with field sample help, conceptualization, and all of my methods, and then UNH COLSA, DBS, and SMSOE as a whole. I also want to thank all of my friends that I've made along the way here. As many people know, I've had a really hard time while being here, and I couldn't have gotten through this without all of my friends that have been here to kind of pick me up and keep me going through along the way. Here's just some of them. There's more friends than this. Also, my family. I was lucky enough to gain an additional family throughout my time here as a PhD when I married my husband, and I have the best in-laws that a person could ever ask for. Everyone is the most supportive group of people that I've ever met in my life in this family, and so I could not do this without them. Also, my dogs, right here. And where's oh, and then them right there. They cause me an insane amount of stress, but I still love them dearly. And then we have a special guest here today, who let me share her birthday to finish my PhD. So if we could sing happy birthday to my little sister really quick, that'd be great. Okay. Happy birthday to you. Happy birthday to you. Happy birthday, dear Lex. Happy birthday to you. Thank you, everyone. And with that, are there any questions? Yeah, Brittany. Hi. Thanks.
I was wondering about the gene expression chapter.
Yeah.
And whether or not you think some of the differences between the cultivated versus the natural population is due to them living in different environments.
I think it definitely could be. I think more studies, focusing on how well mixed Great Bay is would be helpful for answering that question. We've traditionally thought that everything everywhere in Great Bay was traveling everywhere and moving at the same exact speed and settling everywhere. We're not seeing that as the case now. So I think that definitely could be a possible explanation.
So you said you were expecting the restored site to be more similar to the commercial oyster.
Yeah.
Do you have any thoughts about why they might have a distinct
I have no thoughts on that right now. We did not expect to see those results at all. I expected on the admixture plot that I showed where all the different clusters were different bars. I expected to see a pretty even split, like 50-50 of blue and red on those. And when the model suggested that we add a third cluster in there, we thrown for a loop. This could be due maybe to the restoration site being around for a while, so we're getting the next generation in, and they're starting to provide their own genomic signature due to adaptations there.
Yeah, and it's gonna be how often are oysters do that? Is it was it one big question that they visit?
There was one big one, right, Brianna? It was one big one in 2018, I think.
Was it from
the uglies? Yeah.
Yeah, it was 2020 and 2020.
Okay. Yeah.
First of all, I thank you for explaining the nanoform. Been living with her for quite a while and never understood it until I saw your pictures. So thank you very much.
I'm glad.
The other thing is, in Chesapeake Bay, we had like freshwater and saltwater, and we call sweet and salt.
Yeah.
I don't see that here, so. Do you prefer the cultured or the other natural oysters much?
To eat?
Yeah.
I have not eaten a native oyster.
Oh. I don't know. Oh, okay.
Based on the pictures of how they look pretty disgusting. So I'm not sure if I would want to do that. But I do very much prefer cultivated oysters from up here from the south. We were in Georgia not too long ago and we got oysters and they were so sweet that I almost could not tolerate them.
What was that sweet? When you say they're so sweet, what was that?
I'm assuming less saltwater influence. They're in less briny conditions.
Like,
Oh, glycogen. Yeah, I can read it really quick. Okay. They said, are the aquaculture oysters mostly raised from seed in tanks? They're raised and then planted onto the oyster farms, right? Okay. Yes, Dad.
which spot, where most logical, where are they mostly, like where, if you're looking for?
They're exclusively in Little Bay in our estuary system, so there's like a little channel and then a tiny bay area, and all of the oyster farms are within Little Bay, and then the native populations are either within the Oyster River or in the Great Bay proper.
The documentary on down in North Carolina, the Oyster Bay, so I'm just asking, like, what do they need as far as environmentally, what do they need to have a surviving oyster bed and oyster farm? What would you actually need?
The right temperature and salinity conditions.
Yeah, so
They're various, they're
it's different. Huh?
adapted to different areas, yeah. Okay. Yeah.
If I'm personally, would I rather have MSX or Dermo?
Probably MSX right now, as most of the oysters are tolerant to MSX. So you would survive longer and be overall healthier than in Dermo.
I've been tested.
All right. Yeah.
I was wondering about the multiple like experiments that you did in, I think that's chapter
3, yeah.
And whether you explored if there might be an interaction between heritage and the like temporal aspect. So like, you imagine maybe the cultivated oysters might be doing something different with their gene expression.
Yeah, so I did see that in there. So I broke it down into each heritage and then looked at their ability to respond to different collection months or different salinity and temperature, and found there was a significant amount more genes that were differentially expressed in the cultivated comparison than the native one. The native one I don't think had any genes that could find biological functions, and there was maybe only a handful of them, whereas the cultivated ones had I think about 100 genes that had tons of known functions in different oxidative stress pathways.
Between the different
Yeah.
ones. Yeah. So what does that tell you?
That our native populations are not doing a great job of responding to their environment up here. They would benefit greatly if Great Bay was well mixed and we were sharing the DNA between all of them. However, we're seeing the kind of consequences of them not being able to adapt with oyster reefs dying at an exponential rate up here, and the cultivated population is still doing good. So those ones are better equipped for the changing climate that's happening up here. Yeah.
In terms of ecosystem services between the two, are they similar or do the native oysters provide?
The native oysters provide the habitat for juvenile fish. So oyster reefs are in Great Bay specifically are grown in oyster bags, and so they don't act as reef structures and those fish can't hide in there, which also in turn helps with shoreline protection. I mean, we don't see tons of wave energy here, but if there were to be any, those oyster reefs would help more than the cultivated oysters. And they grow longer. Cultivated oysters are harvested at about year 3. And so the bigger oysters are able to filter more. Yeah. What?
I think it was chapter 3, the block beside the volcano, on forget what that's.
A heat map?
The heat map. You compared the heritage as well as the month. The heat map is showing different genes. Is it showing the same genes? Or so it's different genes?
Different genes in each comparison. So each comparison, I identified differentially expressed genes between the two groups, and those just show the top 30 ones that were differentially expressed, just basically to see a visualization of clustering based on gene expression. All right. All right. Well, with that, I will end my talk, and thank you everyone for coming. What? Oh, yeah. You want to take, oh, not me. Hold on. Good job. That was so good. I got to chapter 3, and you know how you were like, once I get to chapter 3, I'm like, what's on it? Thank you, everyone on Zoom. Oh. Hold on.